Welcome to another deep dive. We've got two really fascinating papers to unpack today.
Sounds good.
So the first one is from 2012. It's called A Programmable Dual RNA-Guided DNA Endonuclease in Adaptive Bacterial Immunity.
Okay.
And it basically introduced the world to CRISPR-Cas9, which, if you're following along with the world of genetics, you know is a really big deal.
Huge.
We're going to switch gears a little bit, and we're going to look at long-range incommensurate charge fluctuations in YND Be2Cu306 plus X, which I know it's a mouthful, but it's all about high-temperature superconductors. So are you ready to dive into these two papers and make sense of them with our listener?
Absolutely.
Okay, great. So let's start with that first paper, that CRISPR paper. It's really amazing. This paper reveals this system that bacteria and archaea use to fight off viruses.
Yeah, it's incredible, like their own little immune system.
Yeah, exactly. It's like a personalized defense system. And it's all based on this molecule, this Cas9. Can you tell us a little more about that? What is Cas9?
Well, Cas9 is an enzyme. It's a special type of enzyme called an endonuclease,
Okay.
which basically means it can cut DNA. But what makes it so interesting is that it doesn't cut DNA just randomly. It's guided to very specific spots. And it's guided by these two RNA molecules, CRRNA and tracRNA.
So Cas9 is like the scissors, and the CRNA is telling it where to cut.
Exactly. Think of it like a pair of incredibly precise scissors. And the CRNA is like the GPS navigation telling those scissors where to go.
That's a great analogy.
Oh, yeah.
The tracRNA, what does that do?
The tracRNA is really important because it helps to activate the Cas9. It helps the whole system work.
So it's like the power button.
You could say that.
So this whole system, this is how bacteria can actually remember past infections, right?
That's right. It's one of the most fascinating things about it, actually. They basically take little snippets of viral DNA and they store it in their own genome, kind of like a library of all the viruses they've encountered.
Like a viral mugshot book.
Yeah, exactly. And those stored snippets, they're called spacers, and they're all integrated into this thing called the CRISPR array.
The CRISPR array.
Yes. So when the virus comes back to attack again, the bacteria can use these spacers to create core RNA.
Okay.
And that cRNA will then guide Cas9 to go and chop up the viral DNA before it can do any damage.
That is wild. So can we talk a little bit more about this cutting process? I mean, how does that actually work at a molecular level? The paper mentioned something called a seed region in the cRNA.
Right.
What is that exactly?
So the C region is just a small part of the cRNA, but it's a really important part. It's actually essential for Cas9 to recognize the right spot to cut. So imagine Cas9 is kind of scanning along the DNA, looking for a match with the cRNA. The seed region is like the initial handshake. If that part matches perfectly, Cas9 knows it's in the right spot and it can start cutting.
Okay, so that's the seed region. The paper also mentions something called a PAM. What's that all about?
The PAM, that stands for protospacer adjacent motif.
Protospacer adjacent motif.
It's a mouthful,
It is a mouthful.
but it's basically just a short DNA sequence. And it's located right next to the target sequence. And it acts like a password almost.
Okay.
It makes sure that Cas9 only cuts at the correct location.
So it's like a double check.
Yeah, exactly. Cas9 needs to recognize both the seed region in the cRNA and the PAM sequence in the DNA before it can cut.
Okay, so it's like a lock and key system, but with two keys.
That's a great analogy.
I like that.
Yeah.
So let me just make sure I understand this. If you change the cRNA, you can basically redirect Cas9 to cut a completely different target.
Exactly.
Wow.
And that's one of the things that makes CRISPR so powerful for gene editing. By just tweaking that cRNA sequence, researchers can target Cas9 to pretty much any part of the genome they want.
That's incredible. And this paper actually describes how the researchers simplified this system even more by combining the cRNA and the tracRNA into a single guide RNA.
Right.
What was the significance of that?
That was a major breakthrough. It made the whole system a lot easier to work with, before this discovery. Researchers had to rely on these other, more cumbersome methods for gene editing, like zinc finger nucleases, for example. But this new system, it's much more elegant, much more efficient.
It's like trading your hand tools for a power drill.
Exactly.
Okay, I think I'm starting to get why this paper was such a game changer. But before we get too carried away with CRISPR, we need to shift gears a little bit and dive into our second paper, which takes us from the world of bacterial immunity to the world of high-temperature superconductors. Are you ready for this leap?
Yeah, let's do it.
Okay, great. So this second paper, it's called Long-Range Incommensurate Charge Fluctuations in YND Be2Cu306 plus X.
Catchy title.
Yeah, it's a mouthful.
Yeah.
But before we even get into that, can you just
Yeah.
give us a quick overview of what high-temperature superconductors are?
Sure. So superconductivity, that's this amazing phenomenon. where certain materials, at very low temperatures, can conduct electricity with absolutely zero resistance.
Wow, zero resistance.
Zero.
So that means no energy loss during transmission.
Exactly. It would be revolutionary for our energy infrastructure.
Yeah, no more wasted energy. So what makes them high temperature?
Well, the first superconductors that were discovered, they only worked at these extremely low temperatures. We're talking like close to absolute zero, which is negative 273 degrees Celsius.
Oh, well, that is cold.
Colder than outer space.
Okay.
But high-temperature superconductors, they can operate at slightly warmer temperatures.
Okay.
Although warmer is relative, we're still talking about temperatures below like negative 135 degrees Celsius.
So still pretty chilly.
Yeah, not exactly room temperature.
But still, progress, right?
Oh, definitely. Every little bit helps.
Okay, so this paper focuses on a specific type of high-temperature superconductor called a cuprate, specifically YNDT, Be2Cu306 plus.
Right.
I'm not even going to try to pronounce that.
It's a mouthful.
But what it boils down to is that they're looking at something called charge fluctuations in this material.
Right.
Can you explain what that means?
Okay, so imagine you have a perfectly smooth surface of water, and then someone throws a pebble into the water. What happens?
You get ripples.
Exactly. Charge fluctuations in a material are kind of like those ripples, but instead of water, it's the electrical charge that's fluctuating.
So instead of the charge being evenly spread out, it's sloshing around.
Exactly. And sometimes these fluctuations can actually form these organized patterns,
Oh,
like those ripples in the water forming a specific shape.
So what kind of patterns can they form?
Well, one type of pattern is called a charge density wave, which is basically a standing wave of electrical charge, where some areas have more charge and others have less.
So it's like a wave, but instead of moving up and down, it's a wave of electrical charge density.
Exactly. You got it.
Wow, that's cool. And the paper title mentions incommensurate. What does that mean in this context?
So incommensurate basically means that the spacing of these charge fluctuations doesn't quite line up with the regular spacing of the atoms in the material.
Oh, yeah.
It's like trying to fit square blocks into a round hole.
They don't quite match up.
Exactly.
Okay, so these researchers were basically looking at these strange, uneven ripples of charge in this superconducting material. And they found that. Well, what did they find? What's so important about these charge fluctuations?
That's where things get really interesting.
Okay, you've definitely piqued my curiosity, and I'm sure our listeners hooked too. So we're going to take a quick break, and when we come back, we'll delve deeper into the findings of this paper and explore what these charge fluctuations might reveal about the secrets of high-temperature superconductivity. So don't go anywhere. We'll be right back.
Okay, so we're back, and we're ready to dive even deeper into this world of high-temperature superconductors.
Yeah, these charge fluctuations, they're really fascinating.
They are. So what did the researchers find out about them? Why are they so important?
Well, the big discovery was that there's a direct connection between these charge fluctuations and the transition into a superconducting state.
So as they cooled the material down towards its superconducting transition temperature, those charge density waves we talked about, they became much more pronounced.
So as it gets closer to becoming a superconductor, those fluctuations get stronger.
Exactly.
But then, and this is where I got a little confused, once they cooled the material below its transition temperature, those charge fluctuations suddenly decreased.
Right.
So they increase as it approaches superconductivity. But then they decrease once it actually becomes a superconductor.
That's right.
That seems kind of counterintuitive.
It is, and that's what makes it so interesting.
So are these charge fluctuations helping or hindering superconductivity?
That's the big question, and there's no easy answer.
Okay.
There are a couple of main theories. Some researchers think that these charge fluctuations could be a precursor to something called a competing order.
A competing order.
Yeah, basically something that suppresses superconductivity.
Oh.
So they see that sudden decrease in charge fluctuations below the transition temperature as evidence for this competition. Like, the superconductivity is pushing those fluctuations out of the way.
So it's like a battle for dominance within the material.
You could say that.
But then other researchers think that those charge fluctuations might actually be helping superconductivity.
Exactly. Some think they might be essential, maybe by mediating the interactions between electrons. which ultimately leads to that superconducting state.
So there's still a lot we don't know.
Oh, absolutely. This is still very much an active area of research.
But this paper is adding another piece to the puzzle.
Exactly.
It's helping us to understand the complex interplay between charge fluctuations and superconductivity.
And that could be the key to unlocking the secrets of these amazing materials.
Okay, so we've gone deep into the world of CRISPR, and we've explored the mysteries of high-temperature superconductors, two very different areas of science, but both with the potential to change the world.
What's really fascinating is how both of these papers highlight the importance of understanding fundamental mechanisms in nature.
That's a great point.
Whether it's bacteria fighting off viruses or electrons behaving in strange ways, these discoveries can open up these incredible new avenues for technological innovation.
Absolutely. So let's talk a little bit about some of those potential applications. Starting with CRISPR. I mean, this technology has already been making headlines, especially in the field of gene editing. But where do we stand now? What are some of the most exciting areas of research?
Well, CRISPR has just completely revolutionized gene editing. It's given us this level of precision and ease that we never even dreamed of before.
Yeah.
And researchers are now exploring its use in treating all sorts of genetic diseases.
Like what?
Oh, well, imagine being able to cure diseases like cystic fibrosis or sickle cell anemia just by correcting the faulty gene.
Wow, it sounds like science fiction.
I know, right?
But it's becoming a reality.
It really is.
And it's not just limited to treating diseases, is it?
No, not at all. CRISPR is also being used to develop new diagnostic tools, to engineer crops that are resistant to pests and diseases, even to explore the possibility of creating gene drives to control populations of disease-carrying insects.
Like mosquitoes.
Yeah, exactly.
Wow, it's incredible how far this technology has come in just a few short years.
Yeah, it's really remarkable.
But of course, with any powerful technology like this,
Yeah.
we have to think about the ethical implications.
Absolutely.
I mean, the ability to manipulate the very building blocks of life raises some serious questions.
Yeah, for sure.
So it's crucial that we use this technology responsibly and ethically.
Absolutely.
Okay, so let's switch gears back to the world of high-temperature superconductors. I'm curious to know what kind of impact this research on charge fluctuations could have on the development of new superconducting materials.
Well, if we can understand how these charge fluctuations interact with superconductivity, it could lead to the development of materials that work at even higher temperatures.
So instead of needing those super low temperatures, we might be able to achieve superconductivity closer to room temperature.
That's the dream, right?
Yeah.
Imagine a world where we could transmit electricity without any loss. At everyday temperatures?
It would be revolutionary.
It would. Think about maglev trains that can travel at incredible speeds, or super efficient computers that don't overheat.
It's amazing to think about all the possibilities.
Yeah.
But it sounds like there's still a lot of work to be done.
Oh, definitely. There are still many mysteries to unravel.
But each new discovery, like this one about charge fluctuations, it brings us closer to understanding these remarkable materials and harnessing their full potential.
Exactly.
Well, I think this has been a really incredible journey. We've explored two fascinating and impactful areas of scientific research.
It has been a lot of fun.
It has. And I'm so grateful to have you here with me to help make sense of it all.
My pleasure.
But before we wrap up, I want to leave our listener with something to ponder. What if we could combine the precision of CRISPR with our growing understanding of high-temperature superconductors? Could we potentially engineer bacteria to produce these superconducting materials? And maybe even unlock new possibilities for sustainable energy?
That's a really interesting thought.
It is, isn't it?
I mean, it really highlights the potential for synergy between different fields of science.
Absolutely.
Who knows what other unexpected connections are out there, just waiting to be discovered.
Exactly. So that's our challenge to our listeners today. Think about the incredible possibilities of CRISPR and superconductors. What world-changing applications can you envision?
Dream big.
We can't wait to hear your ideas.
And until next time, happy exploring.
Happy exploring. It's really amazing to think about, using CRISPR to actually engineer bacteria that could make these materials.
It is. It's a pretty wild idea.
Yeah, and it just shows you how, by connecting these different areas of science, we can come up with these really innovative solutions.
Right. It's all about thinking outside the box.
Exactly. So we've covered a lot of ground today, from these tiny bacterial immune systems to these macroscopic properties of materials. It's really incredible how these things can be connected, right?
It is. It really shows you the interconnectedness of everything.
Yeah, it just makes you wonder, What other connections are out there that we haven't even discovered yet?
Oh, I'm sure there are tons. That's what keeps me excited about science.
Me too. So for our listener who's been on this deep dive with us today, what's the one big takeaway you want them to remember about CRISPR and high-temperature superconductors?
Well, I think the biggest thing is the sheer potential of these discoveries. CRISPR has given us this incredible tool to manipulate genes with a level of precision that we never thought possible.
Yeah,
And high-temperature superconductors, they offer this tantalizing glimpse into a future where energy can flow freely and efficiently.
yeah, a world without energy loss.
Exactly.
It's really exciting to think about.
It is, and we're just scratching the surface, really.
Yeah, I mean, these discoveries are just the beginning.
Exactly. Who knows what amazing applications we'll uncover in the years to come.
That's the best part. So to wrap things up, I want to leave our listeners with a challenge. Now that you've learned about the possibilities of CRISPR and superconductors, what world-changing applications can you envision?
Yeah, think big.
What problems can we solve with these technologies? What innovations can we create? We can't wait to see what you all come up with.
Keep exploring, keep asking questions, and most importantly, never stop learning.
That's great advice. Well, thank you so much for joining us on this deep dive. It's been a real pleasure exploring these topics with you.
Likewise, it's been a great conversation.
And to our listener, thanks for tuning in. We hope you've enjoyed this exploration of CRISPR and high-temperature superconductors, and we'll see you next time for another deep dive.